Okay. So as Pete said, I'm Luke Smallman. I'm one of Matt Williams's postdocs at the University of Edinburgh. And I'll be talking about some of the activities we've been doing that are feeding into Dare UK, specifically on constraining the terrestrial carbon cycle. But we'll also have a bit more of a focus on look at the importance of resolution as well. I'd also just like to thank co-authors, David, Andrew, Vasilis, who are actually the ones who've been doing the majority of the modeling work on this, and I've been kind of working more on the larger scale prior works. So the contents is I'm going to introduce everybody to CARDAMOM and Dalek. So CARDAMOM being our model data fusion framework and Dalek being our terrestrial carbon cycle model. And I'm going to advertise the work we've already done, so specifically on the Dare UK domain, carbon cycle prior, and certainly we're very interested in being able to engage more with the atmospheric inversion groups within Dare UK to begin in some fashion using this output, but also us getting feedback so that we can refine and improve what we're offering. Next, I'll talk a bit more on a UK focus, which is where we'll be considering the impact of resolution of our analysis, which feeds into some of our strategies. And then I'll be showing or giving some demonstrations on our modeling capacities for forests, managed grasslands, and also arable agricultural systems. So CARDAMOM and Dalek, our model data fusion framework. I'll start with Dalek. So this is our carbon cycle of intermediate complexity. And what Dalek does is it simulates the various different live pools, so foliage, labile, which supplies sugars for leaf bud burst in the new year, the roots, the wood, their turnover into litter and soil organic matter pools. But we are doing kind of the whole carbon cycle here. So we start, in a sense, with gross primary productivity of photosynthesis, which is sensitive to the environment around it, so temperature and solar radiation, for example. That GPP is turned over into autotrophic respiration, so that respiration of the plant. The remaining MPP is what's partitioned to those different live pools, or MPP allocation. Then have allocation as well from labile to foliage, and then the plant mortality, so canopy senescence, and then the turnovers of wood and root. And then the decomposition of the denic matter to give us a heterotrophic respiration. Now, as well as this natural cycle, we also have the capacity to represent or to impose fire on systems, so we can have fire emissions of the carbon cycle imposed using remote sense data from satellite, specifically MODIS burned area products, but also forest deforestation activities coming from Global Forest Watch, so it allows us to extract the different live pools and then have residues left behind that can subsequently turn over. Now, each of the arrows that are shown there are controlled by parameters, and what's happening in CARDAMOM, which I'll show in the next slide, is we are retrieving the parameters for all of the carbon cycle models. So each arrow, each flux that we're seeing here is being constrained by the observational information that we're going to be assimilating via CARDAMOM. Now, that's quite unique compared to, say, Joules, which starts with having a plant functional type where they have a single parameter set, whereas we're getting everything constrained by data. So CARDAMOM, so this stands for the Carbon Data Model Framework. And sitting at the center of this, we have Dalek, which I've already introduced you to, and then our model data fusion algorithm, which is an adaptive proposal Markov chain Monte Carlo. And the way that the system works is we take our drivers, and that's things like air temperature and short wave radiation, which I've already mentioned, and they can come from either field data or things like reanalyses, as well as then things like disturbance data. So I've mentioned fire and forest. Here is an example of the forest fraction loss averaged across 2001 to 2017. I'm just going to move this little thing out of the way. So even just with this, we can see quite a lot of interesting spatial behavior where most of the deforestation activities are happening in Northern England and Scotland and in Wales, but then there's not a lot happening across most of the rest of England. So there's very different drivers already at play. On the other side, we have biophysical observations, so things like time series of leaf area, which comes from remote sensing. And again, we see an awful lot of spatial variability. You can see London, for example, standing out as quite a low area. much more leaf area on the west coast of England, and also across Wales, which is a lot of managed grasslands. Much less on the east coast, because we've got a lot of arable agriculture, so it spends a lot of the year having been cut. And then sort of different spatial patterns, actually more climate driven up in Scotland. We then have things like soil carbon, which provides a prior on the initial carbon stocks for the soil organic matter pool. In this case, it comes from the Soil Grids database, which is a machine learned interpolation of field data. And lastly, wood stocks, again from satellite, and again showing lots and lots of spatial variability. Quite interesting though is that we have lots of biomass being suggested for forests in far south of England, which doesn't seem to be part of management at all. It's not being cleared, and that's for good reasons, because there's a very big spatial difference in the types of forest across the UK. So we're ingesting all that information. So what happens? The drivers are fed into Dalek. They generate an estimate of the carbon cycle, initially starting from a random set of parameters. They get fed into the model data fusion algorithm. Those estimates are confronted with observations and their uncertainties, and we use those to be able to update the parameters for every location independently in any given analysis we do. Now, what that gives us is for every location, each pixel in any of those maps is not just a single set of parameters accepted for the model, but a whole ensemble. And it's from this ensemble that we're able to generate our uncertainties directly. So for every map that I show you moving on, we have a corresponding map of uncertainty at pixel level, which we can feed into further analyses. Okay, so I'm going to now talk about the Dare UK grid and what that what we're already able to say about that analysis, and hopefully, I wanted to sort of generate people's interest into engaging with using this output. So I've got 3 different maps for the wider Dare UK grid. One for the net ecosystem exchange, where a negative value is signifying a net uptake of carbon, whereas a positive value is an emission of carbon. In the middle, we've got GPP, so the gross primary productivity, and then on the far right, we've got fire. Now, I've picked these 3 just to give us a kind of spread of sort of a net flux, a gross flux, and then one of our disturbance fluxes. And we can already see quite a lot of information. If we look at NEE first, across most of the grid, actually, it's relatively neutral. But we do have areas of uptake in blue, for example, across sort of Central Europe and down towards Greece. Some big areas in the Eastern US. And what's also, sorry, I forgot to say as well, but we do have some sources. So for example, up in kind of the Norway, Sweden, Finland area, and then also a little bit of Canada. You might not be able to see it so clearly, but there are some little red specks. So the system is generating both sources sinks as well as neutral estimates. What I think is also quite interesting is if we look at GPP, it's compared to the NE, it's much more uniform. So we're getting differences in turnover processes that are driving net source and sink that have been fed into the system, which I hope should be able to help give us a better process understanding when we engage with the atmospheric groups. Lastly, looking at fire on the right, we can see that across most of the domain, actually fire is relatively small flux. It's relatively little influence, apart from right the way down where we're catching parts of the savannas in North Africa, where there's much, much larger fire emissions. You can probably just to say see a few sort of slightly larger fires in Eastern Europe going towards Russia as well. Now, a big question is whether or not, oh, sorry, before I move on, we should also point out we do also have this information not just in map form, we also have time series. So I've just shown the most likely estimate of the each of the NEE evolving over time in teragrams of carbon per year for the NEE, GPP in the middle, and for fire on the far right. But just so that you get just to remind you that we generate these uncertainty estimates, I've put in the mean annual flux for NEE and for GPP and for fire down here at the bottom. where we can see the system suggests that across this domain, we're most likely a sink of some 1,400 teragrams per year. But actually the uncertainties, and this is just looking at the 50% confidence interval, is sufficiently large that we are well and truly encompassing both the source and sink ranges. Now, this isn't really very surprising because so far we've given it, as I showed you, leaf area information, one estimate of soil carbon, and also just one estimate of wood stock. So we haven't really told the system much about the trajectories. But we can already see that there's a trend towards an increasing sink strength that's been suggested. If we look at GPP, we're getting a trend to an increasing carbon uptake in photosynthesis, which would be consistent with this decreasing trend for uptake. And it's also consistent with the fact that across this domain, there is a net average increase in leaf area as well as carbon dioxide increasing over time, driving CO2 fertilization. What's also quite interesting, but we've not really looked into too much in terms of diagnosing where this is happening yet, is that there seems to be a reduction in fire emissions over this period of time, ending in 2017. Obviously, it's quite variable, but as you can see from the numbers, got around 140 teragrams, for example, at the beginning in fire versus about 22,000 for GPP. So fire in this over this area is a much smaller player, but still quite important. So we have the question then of, are these priors in the first instance actually any good? Are they kind of useful and meaningful? So the next slide is going to overlay estimates of independent estimates for each of these different fluxes. So I'm going to take a bit of time to talk through them because there's going to be quite a bit of information there. Now, on the maps, we see this stippling has appeared. Now, what we're showing here is that areas with stippling are places where the uncertainties generated by CARDAMOM overlap either the uncertainties of the independent estimate, if they have it, or of the estimate given in the independent estimate, if you see what I mean. So again, looking at NEE first on the left, we can see across the entire grid, barring a couple of places in the US, we've got this stippling showing that our uncertainties and the independent estimates are overlapping, are statistically consistent. So that's actually really quite important, even though the uncertainties are quite large. Now, if I look at the time series, I've overlaid the two different independent estimates that we've got for that. Both come from the Carbon Tracker group. The first one is the CTE, which is their main output. And that's just they just have a single value that's showing the evolution over time. It's actually a stronger sink than what CARDAMOM is suggesting. The CT-SAM, so that's the Carbon Tracker South America, is effectively the same analysis, but they swapped out different priors for fire and for the biosphere, as well as adding in additional tropical atmospheric observations. And that ensemble that they're able to generate from swapping in and out the different priors gives them this uncertainty range. And by adding in the extra observations, what we've seen is that actually it's pushed this equivalent domain to being towards being neutrality, but still with a large uncertainty comfortably spanning 2,000 teragrams, 2 petagrams a year, plus or minus of the source sink line. So quite a lot of uncertainty in everybody's estimates there. So we're really looking forward to being able to reduce that as part of DARE UK. Next, in the middle, we've got GPP. Again, across most of the domain, we've got good consistency. The US is showing up as somewhere we've got some disagreement and a little bit kind of in Eastern Europe and up into Russia. But certainly the UK, where we've got our main focus, and across most of Europe, we've got very good agreement. The comparison I've used for this is FluxCom, which is a machine learning derived estimate of gross prime productivity. Now, I've been a bit harsh in using this as the comparator because FluxCom is particularly well known for not varying over time. It doesn't have a strong trend, whereas more process-oriented models such as Dalek tend to see a much stronger increase in GPP due to increasing leaf area, but also CO2 fertilization which is something that's completely missing from FluxCon. There isn't any CO2 fertilization there. The truth is probably going to lie somewhere between these two, though. And then last, we've got fire. And I've got two independent estimates here, one of GFAS and the other GFED. These slightly different approaches. And that's obviously much, much harder because we can see there's much, much less stippling in the map. But this is quite interesting because if I produced a bias map here, you would see that the bias is very, very small. So fire presents this very interesting challenge where you can have very, very small fluxes that generate very, very low uncertainties that make things look like they are not consistent, even though the differences are very small. So we'll be working on how best to represent and the evaluation of disturbance fluxes going forwards. But certainly across the whole domain, our estimates are quite in keeping with what's happening with GFAS and GFED. And if anything, that probably means that all of our estimates are a little bit of an underestimate because there's increasing evidence coming out that suggests that both of these other products are underestimating fire emissions. So we're in good company. So largely speaking, our prior estimates are actually consistent with independent estimates, which I think gives us a very good basis for moving on into the rest of the project. Now, as I say, a but, there's always a but. We have the question of what is going to be the impact of the choice of our resolution. Now, at the minute, this has been done at a ½ degree and spatial resolution. I'll just move this out the way, spanning 2001 to 2017 at a monthly time step. I should say for the inversion groups, we do have ways of downscaling this monthly time step to sub-daily. So don't let that put you off. So I'm going to move on now, talk the UK focus and how resolution is impacting our analysis. Now, you'll have already seen these maps on the title slide, but what these are is an estimate of net biome exchange from 4 different cardamom and Dalek analyses run at 4 different resolutions. And I've given this in kilometers and degrees resolution just to help people get an idea of what sort of scales we're talking about here. This analysis was just done over Great Britain, and we can already see quite a big influence spatially in terms of the information content going from 1 degree down to 5 kilometer resolution. At 1 degree at 111 kilometer, it's comparatively uniform fluxes. It's quite a narrow range that we're seeing. As we move down, we start to see more extremes of fluxes. So over East Lothian, there's a particularly strong carbon uptake, but nothing really sort of in the corresponding area on Western Scotland. We move down again to 1/4 degree. The strong uptake that was there in the previous resolution is now gone in East Lothian, but now we've got a strong uptake being estimated in the Western Scotland. And then again, if we move on to the 5K, we get this just great depth of information there. You can see the spatial patterning for upland areas coming out. We can now see London, for example, pulling out. Obviously, as an area with very low LAI, it's going to have a low biospheric exchange going on, or low magnitude, I should say. So on this basis, we can see a huge level of detail, which is going to be very important when we're talking about, for example, things like agriculture and managed grasslands. But what does this mean in terms of the national budget? What does this mean in terms of national carbon accounting? I've now taken those maps and I've turned them. So it sounds like someone was taking a breath out. I'm sure I was going to get a question or not. No, okay. I've turned those maps now into a time series for each of the different resolutions. And what we're able to see for, on the top row, we've got is gross primary productivity and then ecosystem respiration, our eco. So those are the 2 big natural fluxes. And then on the bottom, we've got fire and carbon loss from forest removal. So our 2 disturbance fluxes. And if we look at the natural fluxes first, we get convergence when we get towards a ½ degree or less in terms of resolution. That's quite important because that gives us good confidence that that larger prior that we're doing, covering a much larger spatial area, for the big fluxes, for the big gross fluxes, actually is probably quite robust to be done at a ½ degree resolution. But when we're looking at the detail of what's happening, for example, over the UK, which is the core focus for Dare UK, we can see that that's not going to be enough. At each resolution change, we see, first of all, there's an increase in the amount of carbon being lost through fire and forest loss, but also, particularly on fire, very big differences in the temporal dynamics, depending on where the averaging has had influence to interact with carbon stocks and the fire emissions, for example. We get peaks appearing and disappearing. There's a similar, but maybe not quite as clear story in forest losses that are going on as well. So we get we don't get a convergence even down to that 5 kilometer resolution. Now, this is important because at 5 kilometer resolution for cardamom, that's a huge amount of computational effort required. And it's pretty much prohibitive to go any finer than that across the whole of the UK, which means we need to develop strategies that will allow us to include that finer scale disturbance information, but without having the additional computational cost of literally running the analysis at finer scales. Now, I know I said fire and forest are nationally small, but they did increase with progressively finer spatial resolution. Our main strategy for being able to include that finer scale resolution information, particularly around disturbance, is going to be based on tiling. So we're going to pick out each of the different ecosystems below those 5 kilometer grid cells and average their information so that we have a 5 kilometer grid cell that just represents the forest of that area, just represents the grasslands, just represents the agriculture. And this is some work that David Miladowski is going to be leading. He's already made quite a lot of effort in generating the data files, and we're hopefully going to be running our analyses, our first analyses, starting with forests fairly soon. Now, as I've already kind of mentioned, this analysis didn't include grassland management or arable agriculture, which leads us to an important question of what capacity do we have to simulate these systems? Now, to look at forestry, we've actually published on forestry a number of times over the years, but I'm just going to sort of reiterate some of it with one of our recent analyses. We've calibrated Dalek at a range of different forest sites across a number of different latitudes, and I'm just going to show an example of the NEE and the LAI from 3 of those. Got Huhtila in Finland, which is a boreal forest. We've got Le Bray in France, which is a sort of a warm temperate forest. And we've got Harvard Forest in the US, which is partially there because that is the sort of the classic benchmarking broadleaf temperate forest used in the land surface modeling community for people to work with. So we've got in green is Dalek. I'm just showing the median estimate, our most likely estimate. I'm not showing the uncertainty. We've got in black is observations which were assimilated for calibration. And then in blue is evaluation data. So for each of these 3 sites, we calibrated off of the first 5 years of the analysis, and then we projected forwards for however long was left in that analysis, which is why these are also just put down as an analysis month, because they're not overlapping time periods. So just wanted to keep this idea of progressing in time being the key thing. And what we can see across each of the different sites is, even though these are quite different ecosystems, and we do quite well. So if we look at the leaf area and the NEE at Huhtila, we're getting the increases in LEI and the decreases relatively well. We are getting the magnitude changes in sort of between 1 and 4 meters, and a similar situation for NEE. Le Bray doing quite well, but it's a less good data set, so actually things are a little bit more noisy. And the uncertainties are larger in the observations. So we're still performing quite well. And then for Harvard Forest, sort of the key sort of touchstone forest site, we're getting this very good, consistent LAI pattern, which is both observed and what the model is producing. And even though this LAI is actually, it's peaking at roughly the same value every year, there's quite little interannual variability. We can see when we look at the NEE, there is a lot of interannual variability, and critically, the model is getting it. Even beyond the black where we've calibrated into the projection, we're getting these low peaks and sort of the more extreme high peaks over time. which really I think shows a lot of the skill that the model has got. If we move on now to pasture, we've got a version of Dalek which has been modified to include grazing activity, which then introduces disturbance of the system, adds litter fall from the grazers being obviously imperfect consumers, and carbon dioxide and methane emissions from the grazers. We're drawing in information from the agricultural census that gives us knowledge over what grazers are dominating in given areas of the UK and what stocking densities they tend to occur in. That allows us to be able to impose this very sort of changeable and variable disturbance. Now, this version, the model's been calibrated at a site in Midlothian in Scotland. Using a similar concept, there's been a calibration over the first few years where Dalek is the green line for LAI on the top plot and there's NEE on the bottom plot. And the red markers are Sentinel-1 LAI that's been used as calibration. And the red is the eddy flux tower used as calibration for NEE. And similar with the forests, when we're projecting forwards, we see this very, very good capturing of the interannual variability. And in this case, we've got the shaded area, which shows the uncertainty from the Dalek analysis and how we are capturing those observations very well, both for the LAI and for the NEE, and also, again, capturing a lot of the interannual variability. And you can see if we plot that as an XY, getting a broadly good sort of linear fit. Now, if we take that temporal information and aggregate that to the totals, we see for the net ecosystem productivity, so again, the net carbon exchange, in this case, a positive value is a net uptake, and the ecosystem respiration, and then the gross primary productivity. That the model in black is very much getting the variability between those fluxes and very much closely matching the observations, the measured estimates within their uncertainties that are shown in gray. So for this, again, for pasture systems, again, getting a very good fit. Lastly, moving on to crops, we've used a version of Dalek called Dalek crop, which is assimilated again Sentinel-2 LAI information. In this case, it's just information from March and April, which is then projecting forwards roughly 5 months towards harvest. And the model is then estimating the yield. And we're then comparing to observational constraints. So this is in 2 individual fields of winter wheat in the Scottish borders. This is the observed harvest as estimated from the cabin data of the combine harvester going across the field. And we can see a lot of spatial variability with lower areas, for example, in the northeast of the field here, and a higher yield further in the south of this upper field. If we look at how the model's done in projecting and predicting that, we're getting broadly the same behavior. So there's a lower yield being estimated in the north side of the field, higher yield in the south side of the field, and then a bit more challenging, but again, broadly speaking, the same spatial pattern being found in this southern field as well. And this is moving beyond kind of the sub-pixel variability that we're showing at the UK scale. This is now looking inside of actual fields. So we're able to really get at this sort of spatial granularity information with novel data sets coming from Sentinel-2, for example. And again, if we compare that in more of an XY sort of format, we can see getting a very good sort of broadly linear agreement between the observed and the modeled yield, getting R squared of 0.7 there. So we're demonstrating a good degree of skill across a whole range of different ecosystems now. So just to kind of summarize, carbon cycle prior is consistent with the independent estimate. So we're ready for engagement with other partners within DARE UK and to begin doing, for example, forward analyses of our emissions and also using the information hopefully as part of as a prior for atmospheric inversion. The UK focus analysis is demonstrating that actually the natural fluxes are converging really below about a half degree or below or equal to a half degree, which gives us good confidence that the numbers we're getting in the larger scale prior for the gross fluxes are going to be pretty robust. Disturbance, that needs finer scale resolution, which is going to be leading into our tiling approach that we're going to be using to connect things together. And agriculture, both the grazing and also arable agriculture presents a unique challenge, but actually we're developing methodologies that are more than capable of rising to the challenge of simulating those very effectively. So I'd say we developed those systems. So that is everything that I had to say, and I think we've just about stayed on time there. So I'm just going to stop sharing and let Pete take back over again.
Luke, that was perfect timing to the minute. That was fantastic. I would suggest if there's any pressing questions which don't want to wait half an hour, we could ask them now. Anything which will wait half an hour, we'll have a general discussion at the end. So are there any questions for Luke that can't wait?
I have a question. I've forgotten how to put my hand up on the thing. Can I ask it?
I think you can ask it.
Thank you. Yeah, great presentation, Luke. It's really exciting to see all these developments that they made. So I just wanted to ask a little bit more about the harvest stuff that you guys are developing. I mean, the first thing is, are you confident that it's going to scale? Because you've obviously got very tiny scales there. Is that going to scale to UK size? And also, what information are you inputting about the timing of the harvest, or does that matter so much?
So in both the pasture and the arable systems, those are now undergoing testing at sort of UK scale. So we are fairly confident that is going to work. Matt can correct me if I'm wrong on that. And the central information is available across, obviously across the whole UK and increasingly long time period now. So that's also given us good confidence. It seems to be very robust. Lastly, on the point about harvest, again, Matt will correct me if I'm wrong, because this is Andrew Revels, his lead on this. We're using Sentinel-2 because it's so fine spatial resolution and so sensitive. You can see the decline in leaf area that comes with harvest, and you can use that to fit the model. Do tell me if I'm wrong there, Matt.
No, you're spot on. I can just say in terms of a national test, we're just running across all of UK improved grasslands and analysis. And we're actually using remote sensing to distribute livestock across the UK. So we're actually figuring out where the cows and sheep have to be in order to get the time series of leaf area that we see in sampled arable fields. And the first statistical tests are showing good significance. You know, we're putting animals where they should be, and therefore grazing realistic amounts of biomass. And for the arable crops, we're working on getting more data to do evaluations more broadly. But we're confident. Yeah,
nice one.
Any more questions for Luke? Just straight away. If not, I'll suggest we hand over to Silvia and she can start sharing her presentation. Silvia, are you ready?
Yep.
Then take it away.
Yep. Okay, thank you for having me here. We're going to change tack a bit because, so first of all, to introduce myself, I'm a scientist at the Max Planck Institute for Biogeochemistry in Jena, Germany. And what I am going to talk about is a bit different, both in scale and in the focus than what Luke just talked about. I want to talk about the effect of nutrients on the global carbon sink. Before I start with the science, so I'm going to talk about the Quincy model, which we have developed here in Jena over the last several years. It's a land surface model. It's, of course, a huge effort. It's not just me. It's a project led by Zenke Zele, and all these people have contributed. I will try to acknowledge people as we go through, but any work is mostly teamwork. So the science. Why am I talking about nutrients? Why do I want you to care? If we think about the carbon cycle just through the carbon perspective, things look pretty simple. We increase the CO2, that increases photosynthesis, which increases plant growth, which eventually increases the sink in terrestrial ecosystems. And it would be really great if things were like this. In reality, ecosystems are a lot more complex, and there are a lot of factors that contribute to the CO2 effect on the carbon sink. And we can talk a lot about how much complexity we want to add in the models. Of course, we can't do all these things on a global scale. But what I actually want to focus on is nutrients, because plants don't just need carbon to grow, they need a bunch of things, macro, micronutrients. In models, we mostly talk about nitrogen and phosphorus. We've sort of started talking about things like potassium and so on, but let's stick to nitrogen and phosphorus. If they don't have these things, plants cannot grow, they cannot function. So we cannot actually completely ignore nutrient availability in our models. And in fact, this is something that we see if we go out into the real world. So there have been a lot of experiments where people have increased atmospheric CO2 either in free air enrichment experiments or in phase or in chambers. And we see a huge variety of responses. So going all the way from what we would see just with the just if we're thinking about the carbon perspective, all the way to just no response at all. And in fact, this is a kind of older figure. We are missing the Australian phase experiment, nuke phase, where they just see flat out nothing. So there's a lot of stuff going on, and there are a lot of factors that differ between these different experiments, but one of them is definitely nutrient availability. Does this actually affect our model predictions? Yes, it does. This is a figure from a study that's getting a bit old now, so it's an simplified model ensemble where they've added an empirical nitrogen and phosphorus limitation. And what we see in time is a decrease in the carbon sink due to nutrient limitation. Now we've moved on a bit from when this paper was published. So the SIMP5 models, I think only a couple of them had the nitrogen cycle. The SIMP6 models, a lot more do. So we can now have a more in-depth analysis than just this simple factor. But also, even this simple factor shows us that this is something that we need to take into account. And we don't only need to talk about whether we do include nutrient cycles in our models, but also how we include them. So these are results from a study from my colleagues from last year. And what we see in some ways it's very usual. So we have a time series with two ICP scenarios, but the colored areas are process uncertainty. So this is not an ensemble of different models. This is not parameter uncertainty. This is the same model where they have changed the way that we represent essential nitrogen cycle processes. And we see a huge spread. So these are all ways in which different models do represent the nitrogen cycle. And this leads to uncertainty. So as well as saying, yes, we do need to include nitrogen and possibly phosphorus, we really need to say how. So this is sort of what I'm going to talk about. So how? Traditionally, the models are based on Liebig's law of the minimum, which was, I think, started in the 17th century for agricultural plants. So what it says is if plants have a bunch of resources, in this case, carbon, nutrients coming in, they have a requirement based on the C/N ratio or the NTP ratio, and then whatever is limiting, whatever is not enough is just being thrown out. So that's why it's the law of the minimum. So the plants cannot do anything. This is just what's happening. If they're nitrogen limited, they just throw the carbon away. They don't grow. This is not true. From observations, we know that plants have the capacity to adjust to nutrient limitation. So they can do a bunch of things. They can try and get more nutrients from the soil. They can change the way they use the nutrients they do have, and they can change their actual nutrient content, so the stoichiometry, which I'm going to talk about lots of the C/N to P ratio of the tissues. So we do need to figure out how to put this into models. And to answer some of these questions, we have built the Quincy model. We decided to start from scratch to get rid of some of any legacy code, and especially legacy science that might be in the older models. It's a land surface model. It's quite a bit more complex than Dalek, which has its advantages and disadvantages. It has all the things that usually land surface models have. We have a detailed representation of the canopy, so a layered canopy, and also of the soil, layered soil. We have included a bunch of new functionalities, such as the ability to track isotopes, so we can make use of isotope data when we're trying to improve plant physiology and also soil representation. And another thing that we have done this is very much a modular model. So if you've ever tried to go into a land surface model and change anything, you know that this is generally a pain. So it's not very easy to say, oh, let's just try and change how we represent this process. So with Quincy, this is now a lot easier. It's relatively easier to go in and change something and really play around with it and test hypotheses. And this is exactly what I've done. So here are some results from my work, what I've done over the last few years, which is to look at how plants can alter their stoichiometry. Why do we think that they do this? So if there are not enough nutrients, maybe the plants can do the same things with less. And this is something that has been observed. And traditionally, we started with models that have fixed stoichiometry, as I said before, and nothing happens. But then once we figured out that maybe we do need to include this fixed stoichiometry, what models and what a lot of models have right now is a sort of empirical formulation. And what it says simply, if there's not enough nitrogen, we decrease the nitrogen content. If there's too much nitrogen, we increase the nitrogen content. And there are these empirical functions which are based on observations, but the actual shape has nothing to do with plant physiology. And this generally works okay. I'll show you cases in which it doesn't. But what I actually set out to do is to just go a step further and say, but what do nutrients actually do? What does nitrogen do? So I'm using the optimality hypothesis, which says that plants change their function and structure in order to maximize growth. And what this means for stoichiometry is we have to think about the function of nitrogen, which is mostly in enzymes, which in leaves, it translates into photosynthetic compounds, so chlorophyll and rubisco. So it means if you increase leaf nitrogen, you increase photosynthesis. But at the same time, if nitrogen is linked with enzymatic activity, we also increase respiration. So if we look at the black curves here, what it says is, okay, as you increase nitrogen, you're going to increase photosynthesis, keep on increasing it until a point where the respiration starts kicking in and the curve starts going down. So the optimal nitrogen content in this case are these triangles here where the curves starts flipping. But then if we move away from just photosynthesis and we think about the entire plant, if we increase the leaf nitrogen and the tissue nitrogen all over the plant, we also increase the nitrogen demand. So for given nitrogen availability, we're now looking at the blue lines. The more you increase the leaf nitrogen, the less growth you're going to get because there's no more nitrogen. So actually, what I term to be the optimal leaf nitrogen is the intersection of the curves, so the black dots in this case. And the actual optimal nitrogen changes if we increase CO2, it changes if we change the nutrient availability. So if we move in space to somewhere else, or if, for example, there's an increase in nitrogen deposition. So the model can calculate this at every step. It's not a complicated algorithm. It just chooses which way to go. So, okay, we've come up with a new way to represent leaf nitrogen. How do we test it? One option is to just go big. Okay, we're going to run this model for the globe or for Europe. We've got a lot of large data sets. Let's just see how it does. But if we do that, we really lose our capacity to diagnose what's actually going on inside the model. It just becomes very complicated. There are a lot of factors that come in. So what we're going to do here is we're going to start with a small experiment. And then once we've checked that the model works and we understand where it does or where it doesn't, then we're going to go on to the bigger scale. So all the results I'm going to show you for the next few slides are simulations at the Duke Face experiment, which was a experiment in the US in a loblolly pine plantation where they increased the CO2 over several years. So what happens if we do this? So in red you have the observations. In gray is the model that does not change the stoichiometry at all. In black is the empirical model, and in orange is my new formulation with the optimal leaf nitrogen. If we look here, first if we look at the leaf nitrogen, what the data shows, it's a gradual decrease in leaf N content. And this is because, as we were expecting, you increase CO2, there's more growth, more nitrogen demand, the plants run out of nitrogen. So they're starting to decrease the nitrogen. Of course, the model with fixed stoichiometry does nothing. The empirical model predicts a decrease, but it's really severe. So it's a lot more, especially towards the end of the experiment, than the observations. And the optimal model is a bit closer, it's still a bit low, but it does a much better job than the empirical model. What does this mean for MPP? Well, so the model that has the fixed stoichiometry cannot reproduce the observations. So the observations say we see a continued, a sustained growth response over the duration of the experiment. If we don't change the stoichiometry, we can't reproduce that. But both the empirical and the optimal model reproduce the MPP. So there's something that doesn't quite make sense here. So why does it get the leaf nitrogen wrong, but it gets the MPP right? And to answer that, we need to move on to another process that's essential to understanding nutrient limitation. And that is how plants take up nitrogen, nutrients in general. This is work done by my PhD student, Melanie Karen. So I could give a whole talk about plant nutrient uptake, but just briefly, there are several ways in which plants that run into nutrient limitation can increase the nutrient uptake. One is to just increase the root mass to get more nutrients. Another one is to increase biological nitrogen fixation if they're symbionts. Another one is to exude, so to just put the carbon into the soil and accelerate and make decomposition faster. But the one that we have decided to concentrate on is mycorrhizal fungi. So fungal associations that help the plants get nutrients from the soil. This is something that we know most plant species can do, but it is very rarely included in land surface models. So what Melanie has done is she has included two different types of mycorrhizal functionalities. One are the decomposers, which simply dump carbon into the soil and explore more of the soil to try and get the nutrients. And the other one are the saprotrophs, which actively accelerate the composition. And these are functionalities that we know mycorrhizae can have. Generally, we talk about types of mycorrhizae, so ectomycorrhizae and diabascular mycorrhizae, but this is very hard to implement in a model. So we've gone for the physiology side rather than the named type of mycorrhizae. So we're now back at the Duke site. I'm sorry for the color change. Melanie has left, so I can't get her to reproduce the figures. Now we see the data in black. The gray model is the one without mycorrhizae. So it's the same as the one with the empirical stoichiometry from before. And then we have the two types. What we see, if we look at the MPP, so the growth, again, one of the types of mycorrhizae cannot reproduce the data, so it decreases the growth response very fast. And that is just because of the amount of carbon that plants would need to invest to make them efficient. And both the model without mycorrhizae and the saprotrophs can reproduce the observed response. However, if we look at the nutrient uptake, we see that the model without mycorrhizae rapidly decreases its nutrient uptake because it cannot get any more nitrogen. While the model with saprotrophic mycorrhizae then reproduces the data. But so, again, how can the standard model, so no mycorrhizae, empirical stoichiometry, reproduce the MPP? And here we can put both of the things together. And you can see that what's happening is it runs out the gray line. So the standard model runs out of nitrogen, it cannot take up anymore, and then it decreases its leaf end too fast, but can keep up the growth response. So it's getting the right answer for completely the wrong reasons. And I think this is the value in actually looking at an experiment like Duke, because if we're just going to look at MPP, NE, and so on, we would say, hey, this model is doing fine. We don't need any optimal leaf nitrogen. We don't need any mycorrhizae. We're okay. But actually, if we start to look more into detail at what's happening, we see that the model is actually wrong. Really, only now that we've done this, can we say, okay, we can choose one of these model versions to go on and predict the longer term change and the larger scale changes. And this is exactly what we're going to do. I'm going to go to the regional scale, so looking at Europe. I'm going to using data from the ICP Forest database, which is a European level network of sites where they have repeated measurements of a lot of variables. The focus is generally forest health, but they have also have data on tissue stoichiometry and plant growth and so on, so we can use it for our purpose. What do we expect to see? So the ICP data goes from 1990 to present. I've got data till 2015. So we expect as CO2 increases to see increasing nitrogen limitation. And if we see nitrogen limitation, we expect to see a decrease in leaf N. And the question is, does this happen everywhere? Can our model reproduce it? And can we attribute it to CO2, or is it something else? Because just from the data, we can't do that. So that's the advantage of having a phase experiment, that we know that the changes that we see are because we pump CO2 into the air. If we go to the real world, there's also the increase in temperature, a lot of other changes, and it's not easy just from the data to attribute this. So here are the results. In gray are the observations, and in black, the first constraint of the gray and the black is the model with the optimal leaf N, as well as microISA. And in the top panel, we see the absolute leaf N to check that we are actually at least in the right ballpark. Overall, we get the leaf N concentration. We're getting slightly too low values in the needle leaf, and that's partially because of the way that we deal with things like leaf age. But anyway, if we look at the change over the last 25 years, what we see is the observations predict a general decrease in leaf nitrogen. It's not very strong, but it is there. And so does our model. And this is generally across the board, across all PFTs in Europe. Now, can we attribute this to CO2? What we can do with the model is run it with a fixed level of CO2. So with the level, I've used the level 1900. And what we see is that suddenly we don't see this change anymore. So in some places we even see an increase with our CO2 just because of increased temperature, increased soil decomposition. So we can say that from our model, the nutrient limitation is driven by this increase in CO2. Now, of course, the next step, which I haven't got to yet because this is work in progress, is to see what this nutrient limitation means for carbon storage, for forest growth, and whether this increases or is alleviated by plant response in the long term. But this, I think, is a very good starting point to show that the model can work at this scale and can reproduce observed nutrient limitation over time. There's a bunch of other questions also to do with nitrogen deposition that we can explore. And before I leave you, you might have noticed I started the talk by saying nutrients plural. And I've spent a lot of time talking about nitrogen. So models are now moving slowly into also looking at phosphorus. There are two reasons why we need to look at phosphorus. One is that a large part of the globe, so mostly in the tropics and subtropics where the soils are older, is phosphorus limited. And here are some results from the Australian phase experiment, phase, which is very phosphorus limited. And what they show is that they increase, the photosynthesis increases, but most of the carbon goes into respiration. So there is no change in growth. Most of it comes out of the soil, again, soil respiration. So it looks a bit like the plants are trying to get phosphorus, but they can't. So really, if we want to actually look at the carbon sink outside of the northern latitudes, we do need to include phosphorus. The other reason is that we have been shifting the nutrient balance of ecosystems through nitrogen deposition. So we've been putting a lot of nitrogen into ecosystems, especially in Europe, but now also in East Asia, a bit less in the US, but quite a bit. But we haven't done the same thing to phosphorus. So there's this idea that we are driving ecosystems that were previously nitrogen limited into phosphorus limitation. So even in Europe, we might start to be looking at phosphorus. How are we doing in models? Not so great. This is a personal opinion, but a published opinion. We started to build models that have phosphorus, and Quincy's doing the same, so I'm not pointing any fingers. But and we've done two things. One is to improve the representation of soil, because phosphorus does a bunch of different things in the soil, and we've done the same. So my colleague Lin Yu has been looking at that, and we have a very complex soil model out. And when we look at vegetation, we've just been mirroring what we've done with nitrogen. So exactly the same process that we had for nitrogen, we've taken them and copy-pasted P instead of N. This is not strictly correct because phosphorus has different roles to nitrogen. So we would expect plants to have a different response to phosphorus limitation. So we really need to work on that. We have started to work on it. I hope that if you hear a talk from me in a year's time, I will be able to tell you more about how we go forward and show some results about this. But I think right now all I have is this opinion. And finally, to conclude, I hope I've convinced you that we do need to pay attention to nutrients, that they make a difference in models. We have the Quincy model, which is allowing us to test different hypotheses and different ways of representing processes, and that this matters if we want to predict the future land sink with any sort of accuracy. And I can't remember if I said earlier, but the Quincy model is available. We have a Git repository. You do need a username and password, but we're very happy to share. The Quincy model is there to be used by everyone. So please, if you have used it, just ask us. And thank you.
Thanks very much, Silvia. That was fantastic. Great. Do we have questions for Silvia?
Just briefly say before anybody comes up with any questions, if people want to turn on their videos, that's really nice, because obviously we're not seeing so many people anymore. Certainly I like seeing all the faces. So if you feel comfortable doing that, then please do.
Silvia, I'll start off if there are no immediate questions from anyone else. You talk about the leaf nitrogen decrease in the ICP data. What basis is that decrease on? Is that on a mass basis or on an area basis? Because I can see if there's a change in allocation, you get less roots because you have more carbon up top. Which way would the nitrogen change? How would you expect it to change? Can you elaborate on that a bit more?
Yeah, so the data is on a mass basis. The model doesn't make a difference because we have fixed LMA, so leaf mass per area. I'm not sure how that would affect, how that would change with the increase in root mass as well. Yeah, I'm not sure at all. But I think the per area versus per mass is a very good question because that changes in the real world. And it's also got to do with leaf structure and different other adaptations.
Yes, I guess it comes down to having that assumption of a fixed leaf mass area ratio, because that's implying several things, isn't it?
Yeah, which is I know it's not correct, and it's something that we do.
Well, no model's perfect, is it? You've got to assume something. You can have some constants in there.
Yeah, exactly. And once you start valuing everything, then it's actually very hard.
Yeah. Matt, you've got a question?
Yes, thanks, Silvia. Really interesting and exciting, challenging topic. We've gone into this a little bit. We have a version of Dalek with a nitrogen cycle called aconite, and we've published a little bit on this. I mean, we've been able to look at flexible stoichiometry and explore some of those trade-offs, a bit like you did with balancing C and N in terms of light absorbing area versus metabolic rate. The place we've found really challenging, or the process, is in autotrophic respiration. When we try and model plant respiration, of course, that's a function of nitrogen. In most physiological studies, there's a very strong connection between nutrient content and respiration. So that's another reason you would expect plants to have an optimum nitrogen concentration, because they'll just start respiring carbon away if they've got a lot of underutilized nitrogen. But we find the models for autotrophic respiration are so oversimplified and not even clear in their functional forms. Our lack of understanding of what drives respiration means we really struggle once we start pushing the model to look at a few non-analogous states and to change variables and CO2 and what have you. The choice of the respiration model is so impactful on our outputs, and we've no idea which is the right respiration model. So my question to you is, have you had any problems with modeling respiration? Have you noticed that it's a big unknown? And if so, have you managed to deal with it? Do you have any advice? So
we haven't had any problems with it. So even before we implemented any sort of flexible stoichiometry, we had the respiration be a function of nitrogen. And we have the same rates in the leaves and the fine root, and then the wood has a different rate, obviously, because it's doing something different. We also have respiration acclimation with temperature. And the other thing we have is that we separate, especially the leaf nitrogen, into structural and function. And it's only the functional one that actually respires. So when I got to implementing the optimal leaf nitrogen, I didn't specifically run into any problems about that. But having said that, I also haven't looked at any respiration data. So I can't tell you if we're getting that right or not. So I can tell you it's not causing problems with the leaf nitrogen, but I don't know if that is correct.
I mean, we explored in nitrogen and carbon space for a plant canopy. We explored where the optimum was, and we could calibrate any sort of respiration model to get the present conditions right and to find the optimum. When we changed the climate or the CO2, the optimum moved around in phase space, depending on the functional form, whether we chose a Reich style respiration model or a Ryan style respiration model or an Atkin style, right? There's a bunch of models out there. Depending on the one you choose, in our studies, you got very different outcomes for optimality. And that's what struck us as a real challenge. No one knows which of these models is right, because they're empirical anyway. They're just based on a bunch of measurements with a huge amount of noise in them. So we felt that's quite a roadblock.
Yeah, I mean, I think I simplified my life by not trying to change the respiration model.
Okay, yeah.
But this is true for everything, including like if you change the canopy structure, and all of that will change your optimality space. And I think to move forward, you do need to decide on one model. And I think there is a lot to look at in the respiration part and in the connection with nitrogen and phosphorus as well, because there is the ATP connection there. So once you run into phosphorus limitation, there are interesting things happening. So there's definitely a lot more work. I'm also wondering about things like structural nutrients, reserve nutrients, because a lot of the data we have is just total nutrient concentration. We have very little idea which bit respires. So I think you're right. All the models we have are empirical, and I think it will actually be very hard to just hit on the correct respiration term.
That's not a problem we're going to solve in the short term, is it? Because we basically just don't understand the underlying plant physiology of those things. The other thing we don't understand is allocation, the root shoot response when you have elevated CO2 and changes in nutrients. And you relied on the Duke data set because it's one of the few that actually does tell us about that, but it's one of the very, very few, isn't it, that has elevated CO2 and elevated nutrients over a long enough time to say anything.
Yeah.
So we're kind of stuck.
But we have to start somewhere, right? So.
But you got to start somewhere. I guess the nice thing about both you and Luke's talk is that you're capturing the uncertainty. You have different model variants with different assumptions in, so you see the wide uncertainty that we have. Because that's the best we can do just now, I think.
Yeah. I mean, I haven't even talked about the change in allocation. We do have that in the model, but it's just.
I was going to ask why you chose mycorrhiza as the thing to focus on rather than allocation, because that would be a simpler, maybe more obvious thing.
So we have the allocation already included. I just haven't shown the results. We have a not quite empirical, but they increase the root mass under nutrient limitations, also under water stress, and that's a pretty standard formulation. There are probably better ways of doing it, but it seems to work and give pretty much a correct response, but it seemed that other nutrient strategies was something that was really missing. And we thought we'd just look into it a bit.
I should remind everyone that questions are open for Luke as well. Don't forget, we're going to have a general discussion.
I actually had a question for Luke, if everything's open.
Sure.
When you show the changes in the spatial resolution, do you think that's to do with Dalek or with cardamom? Like if you just had Dalek without cardamom, would you see the same differences?
It's difficult to say because in a sense, you can't really just use Dalek without cardamom because we parameterize everything based on the few observations that are available in a given location. But I showed the per unit area fluxes for convergence. But if you look at the totals, then you get more challenges, more differences because you're getting issues around aggregating. You're getting different amount of areas at resolutions, and then giving you different amounts of different values essentially in the observational constraints. So I'm not sure that it's really the model as such as you change the resolution and whatever you're going to calibrate it with, what you're training it to has changed. So you're then getting a different imposition, if that's kind of what you're driving at.
Yeah, I was just trying to figure out which of the bits that you throw in is actually causing that change.
Yeah, I mean, I guess so obviously with Silvia, your work, it's really, it's a very, very different strategy to what we've been doing. And I haven't talked about it in this talk because it's not really what was related to DARE. But Matt, myself, David, who are both on the call as well, are involved with a project, an experiment called Complex, where we actually have about 20 or so versions of Dalek with different process representations. So we've been dabbling with some of the same sort of issues that you've had in terms of like how is model structure and representation actually going to be influencing things. So this is not really a question, but it's actually a bit of an offer for maybe we should have a bit of a conversation about the stuff you didn't talk about today around allocation and what sort of the optimality theory is having on those, because we've dabbled in optimality, as Matt has said, including in sort of calibrating things like the Reich model across spatial areas and trying to force the system to give us some kind of meaningful information. So I'd quite like to have a chat with you about that. I realize that is not actually a proper question and maybe not as helpful for the other 19 people in the room.
Yeah, we can talk about that at a different time, but that's cool. I'd be happy to talk about that.
Can I ask a question of you, Luke? In terms of how do we compare these numbers with what are reported nationally as LULUCF numbers? How do they, can you compare them? Because you've got natural versus the human element of it. How do we start to split apart those two parts of the puzzle?
So for forestry, it's more straightforward, particularly when you're focusing on the UK, because while Global Forest Watch is showing any forest removal, obviously independent of whether it's, well, in the UK, most of that loss is going to be down to humans. So that is actually quite a close comparator that we can have. In terms of land, the other land uses, I think, as Matt was sort of saying, because we've got things like the agricultural census, we can start to connect sort of what are the flux associated with things like agriculture and grazed grasslands. And because I think from Sentinel, there's also some data products looking at how different agricultural types, so crop rotations or switching between grassland versus arable, are being mapped by CEH more kind of year on year. We should be able to partition out that kind of information as well. So I think it can be done, but it's probably a bit tricky. The easiest one would be forestry. That's got the most clear direct comparison.
Has any comparison been made with what's currently reported?
I don't think well, certainly for the forestry, we haven't yet. We have previously in some unpublished works and found that we needed to go to higher spatial resolutions because our earlier analysis looking just at the forestry estate it seemed to be misidentifying, or it seemed to have a big underestimate in the amount of removals compared to what was in the National Forestry Inventory estimates and the Forestry Commission's outputs. So once David and I have done the sort of the tiling approach to get the UK stuff done at as high resolution data as possible, we will make some more comparisons there, and I would expect to see a much closer agreement. Sorry, I'm making David do work now.
One of the issues is so much UK forestry is at sub-kilometre squared resolution, particularly those patches Luke was pointing out in Scotland. There's all those little woodland patches. So this is the scale issue. I still think at 5 by 5 kilometres, we're missing lots of activity that's occurring at the field scale. So that's the next stage to go to. So I think we'll find quite a change when we go to high resolutions and use the satellite data to pick out these small scale operations. But that's the science question that we're going to tackle. But it's a good point, Alistair. We should make a better effort to connect up to the CF stuff and make sure, identify consistencies or otherwise.
I guess part of the problem is that a lot of the LULUCF stuff depends on land use change, which happened quite a long time ago, sort of before 2010 certainly. So you're looking at slow disequilibrium in the soil carbon, which you wouldn't pick up with if you're just using satellite data. But for forestry, it should work, I think, in principle.
I had a question for Silvia about timescales, which you just raised, Peter, right? Slow changes with land use. And it strikes me that isn't that the big problem and challenge for nitrogen and phosphorus is the very slow timescales of response. How do we calibrate and test models that are probably going to need 30 years to see a nitrogen CO2 fertilizer interaction?
I mean, the model you can run as long as you want, but what you're actually asking is where we get the data. And that is a bit of an issue. So I think Pete mentioned that Duke is one of the only places where we have these things together. Data sets like the ICP are amazing, but there's not a lot of them. So this is why I homed into that. There's also another database I've been using, which is the leaf delta 59 content over time. So it was from a paper from a couple of years back, and they say they show global nitrogen limitation. I have my doubts, but so things like that where people have put the data together, it's not, I think the problem with data sets like that is it's not so good for calibration or even evaluation. I think just putting the model and the data together then gives us actually more information. So like I showed with actually fixing the CO2 and seeing what happens, is taking out some of these uncertainties and noise in this just collected large data sets. But for that, we have to have confidence in the model in the first place. And apart from that, you're right, there's not very much. I mean, there are some long-term fertilization experiments in Scandinavia, starting in like the 80s, I think. But they're mostly forestry, so they have data on things like biomass increase. So they don't help us very much with the actual ecophysiology part. But I think if we put all of this together, we do get some sort of larger picture.
Any more questions for our two speakers? If not, time is drawing on.